Dear students, good morning. This is my continuation of my MOOC course, um, Molecular Biology and Cell Signaling. This is my fourth lecture. Is peptide bonds and polypeptide chain. Myself, Dr. Mehdi Hayaz Shahi. I am assistant professor at Interdisciplinary Brain Research Center, Aligarh Muslim University, Faculty of Medicine, Aligarh, Uttar Pradesh. Peptide bonds and polypeptide chain, how amino acids are linked and form the protein polypeptide chain. In this contents of this lecture, I'm talking about peptide bonds, direction of the peptide bonds, and disulfide bond, important of amino acid sequence, what is important, and polypeptide chain and conformational restrictions. And then also we have talked about Ramachandran plot. That is very important for conformational changes. And there's the references for this lecture I've taken from Concerned Biochemistry 8th edition for Libertas Trial, and also another book, Journey from Amino Acid to Protein by Satayev and Amro Nasrullah Boyce. Peptide bond. It is a bond which makes among the amino acids, and proteins are linear polymers of amino acids. The amino acids are linked with peptide bonds. They have the bondation, that's peptide bonds. And the structure of protein determine their functions, and also amino acid sequence determine primary structure, that's very important. And how folding occurs and all the function, biological activities of the amino acid and proteins, and they depend on the different types of binding with peptide bonds. Then we have to very important, that's why we wanted to know amino acid sequence and all the peptide bond formations, how they behave like in the solutions. Peptide bond. It is the covalent bond, covalent amide linkage formed by condensation between alpha carboxyl group. Alpha carboxyl group. This is the bond formations, amino and carboxyl group. This is alpha carboxyl group and alpha amide group. Condensation reaction, that is called dehydration reactions, removal of water, dehydration reaction. Water release an OH from carboxyl group, OH from carboxyl group, and H from amine group. This is amine groups. And then they form peptide bond. This is a peptide bond, peptide bond. And this has the lineage CONH lineage. Peptide bonds, also called amide bond. And when two amino acids link together, they form dipeptides. And the product is releasing the water and peptide bond. Peptide bond. How stability determined? Because reaction is favorable, because hydrolysis process breakdown, and the peptide bond formation require energy, and the source of energy is ATP or GTP in the cells, and it is very kinetically stable. And it's very interesting that even more than 10,000 years, this peptide bond is still exists without any enzymatic, if suppose there's no influence of enzymatic reactions, they still exist. This the peptide bond, there's no break-in in the peptide bond. Many amino acids linked with peptide bond, that's called polypeptide, and each unit called residue. If a small amino acid, couple of amino acids are linked together, that's called oligopeptides. And if large number of amino acids are linked to with the peptide bond, that's called protein. Peptide bond naming rules exactly, because when the amino acids are linked with peptide bonds, their naming rules and they names some modification in the names. The replace of INI and 8 like alanine, valine, glutamate, all the 8 is replaced by YL. Last amino acid keeps in original name. Suppose here we have 4 amino acid: lysine, leucine, threonine, and glutamine. And what happened? LYS attached lysine, I and I replaced with YL, in this case leucine, leucine, then thyrosyl, YL attached, tyrosine, IN is removed, and glutamine remains same. This is called lysyl, leucyl, tricyl, glutamine amino acids or peptides. Polypeptide chain also has directions, and they have two terminal. One is N terminal, this is N terminal, and then had C terminal. So means it has directional you can say, and the sequence in NC direction is very important. When we reverse, is a different direction, different amino acids, different amino acids. So means we have very fixed direction or we have directionality. In this case example, this is tyrosine in N terminal, glycine, then phenylaline, and finally leucine in the C terminal. It has very specific direction. Polypeptides has two parts. One part is the backbone, is a main chain. This is the backbone. This is the backbone. Backbone. This is the backbone. Main chain, amide bonds. This is amide bonds. This is the backbone, amide bonds. Amide bonds. This is a backbone, amide bonds. And then we have a side chain. That is a side R groups, R groups, R groups, R groups. And it is variable. Is a variability that to give the diversity of the amino acids. Also, hydrogen bonding potential of the peptide chains. The backbone also contain CO as a receptors and NH as a donor of hydrogen bond. Exception is the proline, because proline lacks NH donor. There is no NH donor, and it is important for secondary structure like alpha helix and beta sheet. Is very important for alpha helix and beta sheets. The molecular weight of the amino acid, average amino acid has 110 Dalton, is DA stand for Dalton. And the range of the molecular weight of the amino acids, protein range is 5.5 kilodalton to 220 kilodalton. This is the range of proteins, normal range. And the unit is denoted as DA or Dalton, and 1000 Dalton is equal to 1 kilodalton. The size of the protein, typical protein in the range of 50 to 2000 amino acids is normally proteins in normal. Example, this is one of the largest protein that's called titine proteins. It is largest known protein available in the body, especially in the skeletal and cardiac muscles. This is very active muscles, continuous like cardiac muscles is very active. That's why it has titine, and also skeletal muscles are very active muscles. And it is made of 27,000 amino acids. It's very interesting that it's made of 27,000 amino acids. Then peptide bonds also has a very unique amino acid that called cysteine, and it formed disulfide bond. Cysteine is to form cysteine bond, SS bond. What happened in this case? It stabilizes the protein structure. That's why they have disulfide bond. This is cysteine 1 at SH group and cysteine 2 is SS2 group. Then they have oxidation process. And what happened? They form by disulfide bond. This is a disulfide bond. And they release 2 hydrogen ion and 2 electron atom. This is the oxidation process. And it is reversible process also. This is reduction. Then they release the cysteine. This is called cysteine and this is called cysteine. This is cysteine. So this is oxidation process, and it's very important to stabilize the protein structure, and also is very common in extracellular proteins. Whatever the extracellular proteins, they are very important disulfide bondations. There's some special type of cross-linked available in the protein that is called non-disulfide links, and it's present in some protein. Example like collagen, it is in connective tissue, give the strength to the connective tissue, and also fibrin is a protein spot. It has ability to blood clotting, help in the blood clotting, and this is also called non-disulfide cross-linked. Polypeptide chain, each protein has unique and precisely defined amino acid sequence. That is, each proteins are very unique and they're different. They have very unique and precise defined amino sequence, and they are genetically determined. Means from DNA to RNA, RNA to the protein. The process is transcription to the translation, and it's the first time 1953 there is a one scientist, Frederick Sanger. They determined the amino acid of the insulin. It's very common enzyme for glucose metabolism, that's insulin, and the first proved that protein has definite or defined sequences, and this insulin sequencing all amino acid are L amino acid. That's very interesting. And the diversity, the lot of diversity in the amino acid in the protein itself. We have today almost 20 lakhs protein sequences known, and each proteins has unique sequences. If any amino acid in the changes or any kind of modification or a kind of normality, they have different functions. Different function because of their small changes. Polypeptide chain determined by the genetic code. Each amino acid encoded by triplet codons like ATG is codon for methionine amino acids, initial codon, also called initial codon for phosphate acid. Then total amino acid 20 standard amino acids and multiple codons for same amino acid. It's possibility that multiple codon for same amino acids. And gene protein relation, the sequence flow from DNA to RNA and protein. This is a central dogma. We know if the sequence of DNA, then we know the RNA. We determine the protein structure, protein sequences. And gene determine the protein sequences, because DNA nucleotide sequences, we know RNA sequences, then we know amino acid sequences. This is a flow, we can know about that. That's why genes are very responsible for different types of the proteins. Importance of amino acid sequences. It is important because it's determine the functions, essentiality, so enzymatic activities, and also binding specificity, which means a receptor is going to bind with the specific. That's why the amino acid sequence is very important. And also it determines the 3D structure. What is the importance of 3D structure? It is giving the protein folding, how protein is going to fold. Then final structure, also known biological function. And also there's a mutation, changes in the sequences happen in the DNA sequences, then proteins also going to change that. The mutation cause disease like single amino acid changes can cause amino acid like sickle cell anemia. Then we have a cystic fibrosis also going to change. This is a disease because of single amino acid change, and that's called disease. And also this is very important for evolutionary informations. Similar sequence give the common ancestry and used for molecular evolution studies if the protein have similar sequences there. Now polypeptide chains flexibility and conformational restrictions. That's very important. How this amino acid or polypeptide chains show flexibility and also some constraint in the conformation. Polypeptides are has flexibility, but still they have some conformational restrictions there. And this flexibility allow protein folding, and restriction give the stable defined structure. That's very important. So both are important. Flexibility is also important because of the protein folding, and restriction is also important for the stable and defined structure. And the peptide bonds has planarity, mean that peptide bond is planar structure. You see this is peptide bond, alpha carbon, this attached with this NH group. This has plane 1, 2, 3, 4, 5 and 6. So this 6 atom on all together, C alpha, then CO, then NH and C. So 1, 2, 3, 4, 5 and 6. So all 6 atom line is same plane. All 6 atom in line is same plane. That is the planar, or can say flat structure of the peptide bonds. And also very interesting that peptide bonds also gives a resonance. Peptide bonds has resonance, means a characteristic between single to double bond. This is the resonance structure of the peptide bond. Resonance structure is a characteristic that is also shows some sort of resonance. This is single bond here, now a double bond is here. this bond single, double, and now this single bond is here. So that's resonance structure also gives some conformational restrictions and also conformational means flexibility is there. Then they have partial double or partial collector due to the resonance of the amide bonds, the polypeptide chain. And the partial double bond characters is very unique. That's why it behaves like single bond and double bond, and its bond length is 1.3 angstrom. Consequence is that no rotation around peptide bond. Because of that resonance, there is no rotation around the peptide bond. There is no rotation around the peptide bond. They have the rotation here, C and carbon alpha and N carbon alpha, but there is no rotation in the peptide bond. Nature of peptide bond is uncharged, it's uncharged, is no charge, and is allow tight packing of the peptide bond and for the particle protein, and also enable global protein structure. Because of that reason, they acquire global structure, globular protein structure because of peptide bonds. Now the flexibility, backbone flexibility, how they acquire backbone flexibility. Bond can rotate on N alpha and C alpha and C alpha and N alpha, but not in the peptide bond. These are single bond free in rotations, enable protein folding into many conformation, and this gives protein folding in many conformation because of that reasons. And there's a cis and trans configuration, the two configuration. This is the trans and this is the cis. This is the favorable configuration because there's no constraint, there's no string hindrance in the peptide bond there among that. But there's a lot of string hindrance in this in the cis configuration. That's why the trans is favorable configurations. Most common and configurable, and cis is very rare, except in the case of proline, X-proline bonds. So normally you find most of the proteins peptide bond in the transform. Polypeptide chains has two angles. They form two angles. It's called torsion angles. Rotation described by bondation of C alpha with N and C alpha with C bond. There is a one N and C alpha bond called phi angle, and then C alpha with C bond called psi angle. These angles provide the shape of the polypeptide chains. This is the C alpha and N. This is called phi, and this is called C alpha and C alpha and C bond, that is called psi. So they give kind of single bond rotations. For this understanding the rotation or different conformation, we study the Ramachandran plot, and it is developed by Gopal Ramasundran Ramachandran. What it's mentioned is that it is showing that what are the allowed conformation of the protein polypeptide chains and what are the forbidden based on their phi and psi angles, and they mainly restrict steric hindrance, restrict the conformations. And what are the strict exclusion principles? Atom cannot occupy same space. Results, meaning angle combinations are forbidden, and almost 75% of conformation not allowed according to the Ramachandran plot. And this is the Ramachandran plot. This is psi angle on the y-axis and phi angle on the x-axis. And this angle from -80 to the 1 plus positive 80, -80 to the positive 20. What is shows this Ramachandran plot? Not all values are possible without collision between atoms. The most favorable region means more favorable combinations are shown in dark green, this dark green. And borderline region, these are the borderline regions. Borderline regions are shown in light green. This structure on the right is disfavored because of strict, and this structure right side is disfavored because of steric hindrance, and that's why this structure favor only in this range. So normally in the light green regions like you can say 60 psi and also minus 65 region is most favorable. Then again we have minus 120 to the 180 or 70, 120 to 170 plus, and minus 60 of phi at favorable region. These are the favorable region, and this is the most disfavored regions. Phi is 90 and psi is minus 90 is disfavored regions. On the right side, there is no favored regions and no conformation, favored conformation of protein exists in these regions. This is the main Ramachandran plot, and that's give the idea that what is the favor conformation and what is not the favor conformation. And that's why the structure on the right is disfavor. This is on the right side is the most disfavor of string class, because this is a string class and this is the not possible of the rotation. That's why this is not favored regions. Polypeptide chains flexibility and conformation restriction. The protein folding. What is important that unfolded protein, there's a random coil, many conformations, and it is high entropy. And this, if the protein are unfolded and random coil, then it has chances of many conformation form, and they have high entropy stage. So whatever the folding requires, overcoming the entropies mean that it is going to be reduce the entropies when their folding required. And what is the important of role of restriction in the folding? When there's a restriction because of the nitrogen amide changes, polypeptide changes, the peptide bond rigidity, that's very important, and angle limits the reduce the possible conformation, because only 25% of the folding the conformation is possible, and this allow the stable unique protein folding. That is very important to allow unique and stability and also unique folding properties of this protein. So finally, we discuss about protein folding, structure, conformation, all this. Thank you so much for listening to the lectures.